Now we are just going to browse about the cell, which we have already discussed in detail in the previous year, that is ninth standard. So there we have studied that a cell is the fundamental structural unit of a living system. So technically we can define it as a speck of protoplasm that is surrounded by a cell membrane which is able to synthesize, grow, and reproduce on its own. So, all the living beings are made up of numerous such cells which vary in size, number, and functions. So, these cells can be either single celled, examples like amoeba or any protozoa, or they can be multicellular like the fungi, or it can be even more complex like the human being cells. The cells are microscopic in nature, they vary in size. They can be as small as our own red blood cells, which are found in blood, or they can be long like the nerve cell, and they can also be a large cell like the egg of an ostrich. So the cells are of varied in shape. The RBCs are concave and circular, while the white blood cells are amoeboid in shape. The nerve cell is elongated, then the stomatas are bean shaped. So these individual cells in a group form tissues, and these tissues organize and perform a single function, that is what we call it as an organ. So these cells contain thus small organelles floating in liquid protoplasm and functions in a greater coordination. The plant cell and the animal cells are entirely different in structure and function differently. If we look at the enlarged diagrams of both the plant cell and the animal cell, one would be surprised to know the functioning occurring at the greatest coordination. The essential part of a cell is cytoplasm. The cytoplasm is the liquid or the living liquid present inside the cell, which contains organelles like mitochondria, Golgi bodies, endoplasmic reticulum, lysosomes, centrioles, ribosomes, etc. So, all these small cell organelles will be floating in the protoplasm or the cytoplasm. Apart from this, many reactions that are catalyzed by enzymes are also taking place in this cytoplasm. Then, in the center of this cytoplasm, we have nucleus, yet another smaller wall where the genetic material, that is the information required for the cellular activities, are stored in it. So it gives information for growth, for division, the formation of proteins, enzymes, and the messages for the reproduction. So if we look at the enlarged diagrams of an animal cell and a plant cell, we would find in an animal cell the outer membrane is typically a bi-layered lipid membrane. It consists two layers of lipids, fats, and these lipid membrane is disturbed by the presence of proteins. Hence, together the cell membrane, we always regard it as a lipoprotein in nature, which has lipids and proteins. Then The cytoplasm has mitochondrion, also called as the energy or powerhouse of the cell. We have Golgi body. Golgi body are nothing but 2 to 3 sacs which are arranged over, and they are the protein releasing factories. Then we have centrioles, the nucleolus in the center. The nucleolus has a nuclear pore and the nuclear membrane. And apart from this, we have lysosomes. Lysosome lodges many enzymes. It is the site of secretion of many enzymes. Then we have endoplasmic reticulum, which are the skeleton of a cell, extend from the nuclear pore to the cell membrane. They are tubular in nature, and if they lodge ribosomes over them, or if we find ribosomes over the endoplasmic reticulum or attached to endoplasmic reticulum, they are called as rough endoplasmic reticulum. And if no ribosomes are found attached to the endoplasmic reticulum, then such a endoplasmic reticulum is known as the smooth endoplasmic reticulum. Apart from this, we have centrioles, which are required for cell division. Their role seems to be more prominent during the cell division. And coming to the plant cell, so the outer membrane is a thick cell wall which is required for the rigidity and provides strength to the plant cell. Similar to the animal cell, we find nucleus, cell membrane, rough endoplasmic reticulum, smooth endoplasmic reticulum, mitochondria, and Golgi bodies. So, compared to animal cell, we have here more number of chloroplast, which are the centers for the preparation of food, unlike the animal cell. So we find chloroplast only in the plant cell and not in the animal cell. The other major difference that we find is the vacuole. The vacuole is a large portion which has water, and it gives the pressure to retain the structure of the plant cell. And coming to the parts that are very essential to both plant and animal cells are the outer membrane, the cytoplasm or the protoplasm, and the central nucleus. But in plants, the outer membrane is covered by a thick coat called as the cell wall for greater rigidity. The outer membrane is also called as the plasma membrane, and this is completely made up of a lipid bilayer as we have studied, and this is semi permeable in nature in animal cell. Semi permeability means it has the ability to allow certain substances in and out of the cell through the plasma membrane, and the membrane is broken by the presence of pores which are protein in nature. The cytoplasm is the essential part. It is a fluid which lodges organelles like nucleus, ribosomes, liposomes, mitochondria, centrioles, endoplasmic reticulum, etc. These organelles are very specialized in their function. Apart from this, the cytoplasm also has soluble and insoluble molecules. And these molecules can either be organic in nature or inorganic in nature. The central part is the nucleus, which stores the information required for growth, division, formation of proteins and enzymes, and reproduction. It is also surrounded by a nuclear membrane broken by the presence of pore, which are protein in nature. Now, coming to the part of the cell and their functions to be studied, we'll start with cell membrane, also called as plasma membrane. Main features are it is the outermost membrane in animal cell. It is lying next to the cell wall in case of plant cells. This plasma membrane is very thin. It is flexible lining membrane. It possesses flat pores. It is semi permeable in nature and it is made up of lipoproteins. And the function of plasma membrane are it separates the contents of the cell from its surroundings. It regulates the entry of certain soluble and compounds and ions. It maintains shape of the cell in animal cells. Next, the cell wall is found only in plant cells. Cell wall is the non-living rigid layer surrounding the plasma membrane. It's mainly composed of cellulose, which is a larger carbohydrate. This is freely permeable, unlike the cell membrane. So, this cell wall allows everything to move in and out of the plasma membrane in plant cell. It gives functions, it gives rigidity and shape the plant cell to the plant cell, allows substances in solution to enter and leave the cell without any hindrance. It provides protection. Cytoplasm. All the parts together inside the plasma membrane, excluding nucleus, is cytoplasm. This cytoplasm contains a mixture of water and soluble inorganic and organic compounds and various organelles. Its functions are varied. All the organelles have their own different functions. All metabolic activities occur in it. Medium of earlier steps of respiration takes place in this cytoplasm. Next is the endoplasmic reticulum. This endoplasmic reticulum is called as the exoskeleton of a cell, which is an irregular network of tubular doubled membranes. It is continuous with the plasma membrane on the outside and the nuclear membrane on the inside. It may be smooth endoplasmic reticulum or rough endoplasmic reticulum depending upon the attachment of ribosomes. It mainly functions as supportive framework for the cell. It synthesizes and transports proteins and fats. The next organ is mitochondria, also called as the powerhouse of the cell, since it stores energy in the form of ATP, that is adenosine triphosphate. So, this mitochondria can be of various shapes, but usually sausage or boat like. It is double walled, inner wall thrown into the form of cristae, and this cristae lodges many proteins, which are the centers for the manufacture of this ATP energy molecule. So these mitochondria do have their own DNA containing several genes, and they also contain their own ribosomes. Release of energy from pyruvic acid in the cytoplasm, and this enters into the mitochondria to generate ATP. And as and when there is a requirement, the cell releases ATP from this mitochondria to the required site. And synthesis of respiratory proteins and enzymes do take place in mitochondria. The next apparatus is Golgi apparatus, and this Golgi apparatus is found in animal cells, and it is called as dichotosomes in plant cells. So these are nothing but stacks of membranous structures, consist of tubules, vesicles, and vacuoles. So synthesis and secretions of enzymes, hormones, etc. take place. The final processing of proteins also takes place in Golgi apparatus, and formation of acrosome in sperm cells takes place here. The next organelle is ribosomes, also called as the protein factories of a cell. So these are very small granules either scattered in the cytoplasm or found attached to the endoplasmic reticulum. They are single walled, dense spherical bodies composed mainly of ribonucleic acid or RNA, and it is the site of protein synthesis. Now, once the message for the protein synthesis comes from the nucleus in the form of messenger RNA, the transfer RNA present in the cytoplasm picks the required amino acids from the cytoplasm, joins them together, and joins them together and takes them to the ribosomes for their protein formation. Next organelle is, next organelle is the lysosomes. Lysosome is a membranous sac which is budded off from the Golgi bodies. It contains many different enzymes. As many as 40 enzymes are being secreted from lysosomes. So it functions, helps in the intracellular digestion. Lysosomes help in destroying foreign substances. When a cell is old and injured, these rapidly destroy these organelles, and hence they are also called as the suicide bags. Digest cartilages during the formation of bones. The last organelle is the nucleus. The nucleus lies in the center of the cell. It is the largest cell organelle, mostly spherical and dense, and this nucleus is filled with a fluid called as the nucleoplasm, and it has an envelope. The nuclear envelope contains a network of fine thread-like structures called the chromatin fibers, which contain DNA, the genetic material. Functions: this nucleus regulates cell functions. It removes, if this nucleus is removed, the cell dies. Contains chromosomes or genes that are necessary for the survival of the cell. And coming to the discussion about the distinction or the differences that we see between plant cell and animal cell. To start with the first feature, cell wall. Cell wall is definite cell wall. This is made of cellulose present in plant cells, but it is absent in animal cells. Centrosome, it is absent in plant cells and it is present in animal cells, and its prominent role occurs during the cell division. Vacuoles, they are prominent, and these vacuoles can be one or more. They are present in plant cells. They are required for maintaining the cell structure in plant cells. And vacuoles, if at all present, they are present in very small number and smaller size in animal cells. Plastids, they are usually present in plant cells. They are the organelles which contain chlorophyll pigments that is required for the synthesis of food in case of plants, and no plastids are found in animal cells. Size, usually plant cells are larger and the animal cells are smaller. Cytoplasm is not dense in plant cell, while the cytoplasm is more dense and grandeur due to the presence of a wide range of specialized organelles, organic and inorganic compounds, enzymes, hormones, etc. are present in animal cell, and hence the denser arrangement of cytoplasm. It's only a thin lining of cytoplasm, mostly pushed to the periphery, while the cytoplasm fills almost the entire cell in case of animal cells. So next here is a experimental evidence at our laboratory level to show for the presence of an cell, or to show the student how exactly a cell looks like. So the required materials would be a onion, a glass slide, a cover slip, and a microscope. So when you cut open the onion, and the inner wall, that is the concave position of the onion, if you scrape, we get a thin, transparent, fleshy material. Using a forceps, collect this or remove this transparent fleshy material and put them into a beaker containing water. Let it be there in water for 1 or 2 minutes, after which take this transparent fleshy material, put it on a glass slide, and cut into a square or rectangular piece, and now add a drop of any colored solution that we find in a small laboratory like yosin or crystal violet, and cover this material with a even thinner glass slip called as the cover slip, and focus it under the compound microscope, which usually a biology lab has in every schools and colleges. So when we focus, we really observe as shown in the diagram. The compartment is nothing but the cell, and the single darker patch that we see in the compound microscope is the nucleus. So a single small piece has been divided or broken into a number of compartments, and each compartment has a dotted like structure called as the nucleus. So this is the onion peeling or the scale or the transparent fleshy structure as seen under the compound microscope. Thank you.